Yeah, so Sally Temple. I am the scientific director of the Neural Stem Cell Institute and also CSO and founding of LUXA. I have worked now my whole career on understanding how the nervous system is built and how it's maintained as a healthy nervous system. And early in my career, I discovered that the nervous system contained a stem cell that actually creates and builds and maintains the nervous system. So my career has really been understanding those very important stem cells and how they make different types of brain and retinal cell, and how we can use them to harness them for therapy development. Thank you for the So through this, I've been very fortunate to have some leadership positions. I was the president of the International Society for Stem Cell Research. That is the largest global stem cell organization. That taught me a lot about the different types of stem cells and how they're being used across the world. I also was fortunate to be invited to give a keynote lecture at an FDA workshop recently in February on the development of cell therapies. I had to cover the past decade and think about the future decade. And I think the message overall from the workshop was very optimistic, that we are in a new era of regenerative medicine, and the regulatory agencies and the science and the medicine coming together is really important now to develop this new way of medicine.
분야 연구의 선구자로 알려져 있습니다. 처음에 어떻게 줄기세포 연구를 시작하게 되었는지 궁금합니다.
When I was an undergraduate, I was very lucky to be at Cambridge University at a time when developmental biology was really coming into its own. And I thought it was very fascinating to understand how organisms develop. You know, how do we have a head at one end, a tail, feet at the other? It's such a very profound question. And the nervous system is the most complex system. So how do we make a nervous system? How do we make these incredibly diverse cells and have them interconnect and function so that we can think and see? And I just think it was such an inspiring question. I wanted to spend my career on it. Yeah. So thinking about how the nervous system forms, naturally you look at the cells that make it, and that's how we discovered the stem cells.
덧붙이자면 이 파이프라인을 가지고 조금 끝까지 회사가 개발을 끌고 나갈 것인지, 아니면 뭐 중간에 라이선스 아웃을 해서 좀 빅파마가 개발을 하게 할 것인지, 이런 좀 큰 틀에서 그런 계획 같은 거를 설명해 주시면.
You know, so many things that we do in medicine, new things that we do, don't work. That there are a lot of failures. So right now, the focus really is on advancing something that really is working, both preclinically and in the clinic. And that gives us a lot of options. Both paths are open, I guess I'll say. Pharma companies now are in licensing almost half of their products from companies like us that have gone through the early stages of taking an idea that may be a wild idea and turning it into something that looks like it's safe and has early efficacy. They have the ability to then expand that dramatically, not just within the United States, but across the world in terms of treating a number of patients. And if our goal is to treat patients as quickly as we can, that's one way to accomplish that. But it's not the only way. We can also go forward on our own, raise additional capital, and run a pivotal trial that gets approval that says we can now actually start selling the product. Either way, we will be bringing in others to help, because if we're going to be selling the product and manufacturing at that scale, that's much larger than we are right now. But the key thing is, so much fails in the early stages that we're getting through with data that looks safe. Thought that might be appropriate to bring up is one of the key challenges for cell therapies is manufacturing. And let me step back a moment on that. Right now, cell therapies are proving out in a number of different areas to have some pretty powerful effects. We've seen data coming out in diabetes with cell therapy. We've seen it coming out in serious inflammatory diseases like lupus, the ability to quell the immune system. Just this last week, there was an announcement about a cell therapy treating Parkinson's disease in early studies. And the data that you're seeing from us and others in RPE cells for treating AMD all show the ability of cells to do things that a single drug can't do. And that's exciting. And it gets a lot of interest.
이제 신경 출리세포에 연구하다가 갑자기 항반 변성 치료제로 이렇게 개발하게 된 어떤 에피소드가 있으면 좀 알려달라고.
The most memorable thing was seeing my mother struggle to read something. It really made me realize just how debilitating this disease is. And it really came together that Jeff has his clinical practice. I saw my mother struggle, and together we decided to try to improve, to use the power of stem cells to apply it to this degenerative disease. As we said earlier, once the cells are gone, they're gone. You can't drug them anymore, right? So we need something to replace them. So it really was seeing her struggle. Yeah.
광수형체가 다 망가진 상황에서 RP 세포만 잘 이렇게 회복시킨다고 해서 망가진 광수형체가 과연 원래대로 회복될 것인가에 대한 의구심, 그런 상황에 대해서 좀 이렇게 확신을 좀 해주셨으면 좋겠습니다.
That's a very important question. First of all, in terms of the skepticism, I would say that this is a very different cell with different properties, and our results so far indicate that it is showing better outcomes. So I think that's encouraging. And in terms of the loss of photoreceptors, the photoreceptors actually can remain there for a long period of time. They can lose the outer segment, but they are not dead. They are sort of dormant. And it has been shown that these dormant photoreceptors can regrow and the outer segment and become functional. So we believe that that is one of the things that is occurring. I would just add one point that Dr. Stern recognized this early and actually put criteria in for retinal thickness, because if the retina is very degenerated, there's no photoreceptors, then again, your point is right. But for a long period of time, the retina is thicker, and at that point, they're rescuable photoreceptors.
So dry AMD is a slow disease. that goes through stages. Photoreceptor cell dysfunction, photoreceptor cells become retracted or dormant, as Sally says, and then photoreceptor cells are lost. We only treat the middle and first, and that takes years. So there's a good window to intervene during that time. And we have specific criteria, as Sally mentioned, in our trial. Enrollment criteria of 125 microns of thickness in the center of the macula, that when the photoreceptors are gone, there's no thickness. So we make sure there are some photoreceptors there before we intervene. And so after maybe 10 or 30 years of dry AMD, the photoreceptors are lost, but we have a long period of time to intervene.
정확한 인디케이션 125mm 두께 정도면 아까 글씨를 어느 정도 볼 수 있는 여지가 있는 건지.
I could say one more,
Yeah.
that 8 subjects is not enough to know the connection between visual acuity and the morphology, the shape of the lesion. So we need more subjects. Why did one subject improve a lot compared to another subject? We're going to find out. Maybe duration of disease, duration of atrophy, duration of dysfunction. We will see.
이제 좀 빠르게 아까 프레젠테이션에서 잘 설명해 주시긴 했지만 이제 건성 AMD 치료제 분야 특히 요거를 이제 줄기 세포 치료제로 개발하는 많은 기업들이 있는데 이제 이 룩사 바이오 테크놀로지만의 차별점을 조금 더 명확하게 설명해 주시면 좋을 것 같습니다.
Let me first say that it's good that there are a lot of people trying to do this. It means the interest is there. And it's also good when they report good results, because that's supportive that what we're doing has a better chance of working as well. But we are different from the others, and the difference is the cell that we're actually using. In their cases, taking an iPSC cell or an embryonic stem cell and trying to turn it into an RPE cell is a long distance, and it's not necessarily easy to do, and it doesn't always work completely. We're taking a cell that's designed to become an RPE cell, and we're encouraging it to further become an RPE cell. So it's a much more direct process, and we believe from the data we're seeing right now creates a better RPE cell than if you start with something that's not an RPE cell and try to turn it into an RPE cell. Maybe adding to that, if you make a cell 90% like an RPE cell, that's different from being 100% an RPE cell.
많은 환자분들이 대체 그러면 언제 이거 맞을 수 있는 거냐, 언제 상용화가 되는 거냐에 대한 질문을 가장 많이 받는데, 이제 시작이 일상이지만 그 일상을 통해서 저 환자분들한테 어떤 희망적 메시지를 준다면 그 상용화의 어떤 기간과 그런 좀 어떤 희망적인 메시지를 좀 같이 묶어서 얘기해 주시면 고맙겠습니다.
Right now, what we can tell people is that we're in the clinic, that it appears in the number of patients we've treated that it is safe, that we're seeing positive results, but it still has to go in many more patients. What we can tell people is that there's hope, that there's cells that are in people that appear to be safe, that are doing good things, and there's hope. One of the things that may encourage us to license our product sooner, as soon as this coming year even, a year or a year and a half from now, is if a larger pharmaceutical company can help us get there faster, such that we can treat patients that much sooner, then it might be a good idea for us to do a license agreement or partnership next year and let them help speed the process up to get to patients that much quicker. So that will be one of the things we need to consider as we go forward.
그래서 하나 더 질문이 있는데요. 그 아까 FDA RMAT 지정 그거가 있으면 조금 더 상용화 시기를 앞당길 수 있는 건지 궁금하고, 그 원래 제가 들은 거는 뭐 항상 ODD 뭐 이런 그런 지정인데 알맷은 처음 들어보는 거라서 그런 뭐 희귀의약품 지정 이런 거랑은 어떻게 다른 건지 좀 설명 부탁드립니다.
To receive an RMAT designation means the FDA acknowledges that we are treating a serious disease, serious or life-threatening disease, and that the current clinical data is promising. There's no guarantee, but that it's promising. So that means the FDA is willing to work with us to expedite the development of the product in order to get it to patients that much quicker.
부탁 아까 영상에 소개 좀 부탁 여기도 소개 좀 부탁해주고 마지막으로 한국 우리 좀 환자들한테 좀 희망을 주는 메시지를 향후 계획 포함해서 마지막으로.
I'm Jeffrey Stern. I'm a research scientist and a physician caring for patients with retinal diseases such as macular degeneration. I work with Luxa to help guide the new RPE stem cell therapy to address dry AMD. The large number of dry AMD patients I see every day ask for this treatment. We're excited to work hard to bring hope and vision improvement to Korea. Thank you.
My name is Keith Dion. I'm the CEO of Luxa. By training, I'm a scientist. I began my career working in cell therapies, initially for diabetes. And it's been many years now, but we're now seeing cell therapies for diabetes start to be able to affect patients. And there's hope for many patients with that disease. But I got excited at seeing what Sally and Jeff were doing with Luxa in terms of using cell therapy to treat patients with dry AMD. And the progress that has been made is amazing at the stage we're at. I would give credit to Y2 for bringing our product to our attention to Korea much sooner than it otherwise would be because of their financing and funding of the company.
So we always envisioned what we were developing as something that could impact people widely, not just in the US and not just in the UK where my mum was, but everywhere. We have now worked with the Luxa team in Korea for quite a few years. We have gained enormous respect and truly have appreciated this partnership. We know the biotech growth that is happening and has happened in Korea that we see as being a wonderful venue for developing cell therapies. And so I agree with you, we have great loyalty and appreciation for the partnership with Korea that we would definitely, always remember that and see that as very important for delivering this therapy and helping to develop the therapy in Korea.
Two of our officers are Korean, and Korean company owns almost half of Luxa. So there's a very strong Korean. Well, there's some go out to individuals, so.
That's right.
But, so there's a very strong Korean connection.
그 전에는 그 혼자서는 할 수 없었던 활동도 이제 다시 할 수 있게 됨으로써 이제 거기서 큰 만족감을 이제 느꼈다고 했을 때 또 환자들이 이제 그 치료받은 눈과 이제 다른 치료받지 않은 다른 한쪽 눈의 차이를 그 이제 생활하면서 이제 직접 이제 비교를 이제 하면서 희망을 갖고 이제 하는 거를 듣게 되는데 이 또한 굉장히 인상 깊었습니다. 그리고 저희가 이제 1년 동안 이제 추적 관찰을 했는데 그 환자 같은 경우는 그 시력 개선 효과가 지금까지 1년 기간 동안 지속됐다는 데서 굉장히 고무적이라고 저는 생각하고 있습니다.
여기 마지막 페이지에 나와 있기도 한데 코호트 지금 1 완료 후에 코호트 2 진행 중인데 연구에 따른 구체적인 성과와 소감에 대해서 말씀 부탁드립니다.
2 patients into the second cohort. And I mean, in general, the process hasn't really changed. You know, the dosage is higher. Yeah, I wouldn't really say there's any, you know, from the first 6 patients to the second patients in the next 2 patients in the second cohort, you know, the dosage is higher, but apart from that, you know, really the surgery is the same, the counseling is the same. Patients do seem, when they learn about the trial, they learn obviously that there are two different vision groups, that they're increasing doses, and you know, sometimes patients were like, well, I would like to get the higher dose or the middle dose. And so I think maybe the change is like that they're happy that they, on the second dose, because perhaps, you know, some patients might feel like maybe there's a chance with the higher dose that there may be some improvement for their vision, although we don't really know that. That's what we're testing. So that's what I would say is the main change. But apart from that, I mean, the whole process has been very similar to the first cohort.